Hi everyone. Before we get started with the lesson on fatty acids today, Seth says hello. Say hi, Seth. And then there's Shaddai. They're going to help me with a little demonstration. And we're going to start out with something kind of weird. We're going to make some Kool-Aid. So we're going to start with a how-to video of how to make Kool-Aid. Let him drink a little bit. There we go. So we've been talking about metabolism, how we turn our sugars, so we have some sugar here, into energy. So this all happens in our body, which is, 60-70% water. So we have our water here. All right, so we have our water here.
Seth is going to drink it.
And in our body, we eat lots of sugar. So pour some sugar in here. So when you mix the sugar in the water, it's completely soluble. Right? So sugar is completely soluble in water. And that's a good thing because it's going into our body and we need to break it down. So what are we going to break it down with? Enzymes.
We're going to break it down with enzymes and then put some enzymes in.
So we put some enzymes in our sugary watery body mixture. Alright? So now our enzymes can work because they're both soluble in our body. Alright. So, but sugars are not the only thing we get for energy. We also use fat. So this is just some vegetable oil. So we use fats for energy.
Don't you do it, Mom.
Okay, so pour some of this in there.
Don't.
Pour it in there. Disgusting. She says disgusting. You probably can't see this, but on the top, as you would expect, it's bubbling. We have bubbles are packets of oil. So we have a layer of oil that sits on the top of the enzymes and the water and the sugar. So even though our body is good at using our water-soluble enzymes, oh gosh, this is going to be nasty, Seth.
Good. You said good.
You don't want that anymore. Enzymes are water soluble and they're good at dissolving the sugars. They're not. Stop it. They're not so. Stop it. They're not so good at dissolving the oils, which separate from the enzymes in the water. All right. So this whole lecture is going to be devoted into how we actually get the fats or our oils digested and transported so that they can be utilized or broken down by our enzymes to give us energy. And I'll just forecast that one of the things, or the first thing that's going to happen, is our body has bile salts. So we got this detergent. We're going to call that a bile salt.
Guys, I'm going to tell you this. This is not how you make Kool-Aid. Do not put vegetable oil or soap in it.
Okay, so now stir it up, stir it up, stir it up.
There's bubbles everywhere. This looks disgusting.
All right, should I tell them what happened to the oil layer that's on the top?
So it kind of make a bubble, and now it's sparkling.
Sparkling. But the oil layer, you can't see this, but the oil layer that was on the top, it pretty much disappeared. So the first thing our body is going to do is take some kind of amphipathic compound. This is detergent, but similar to detergent. An amphipathic compound are bile salts that are going to emulsify the oil. Go ahead. Emulsify the oil so that they can be broken down by our enzymes, our lipases, and all the other stuff, so we get one homogeneous goop of water and salts and amphipathic detergents and enzymes, so they can be broken down and utilized by our body. That's
Put
enough.
more detergent in.
All right. So that is the topic of our first lecture, how we get our fats digested and then transported so that they can be metabolized and broken down into energy. A whole lecture devoted to that. And then our lecture 2 and on next week are going to be how our fats are oxidized. So that's called the beta oxidation of our fats, just like the glycolysis cycle, just like the citric acid cycle. How are our fats oxidized? And then they're going to be carried into the citric acid cycle so we can get energy out of them. And then also in the next lecture, we're going to talk about ketone bodies. We're going to refer to things like the keto diet. Well, that comes from how your body goes through ketosis and turns our fats into energy if we're in a situation where we don't have enough carbohydrates around. So that's what you have in store for the next 2 lectures, how we get from fats, break those down, digest them, emulsify them, transport them in our bodies, and then how they're oxidized, basically how we get them into the citric acid cycle, and when we can't, under certain conditions, how we turn those into ketone bodies and then get energy out of that. Just a quick list of facts about the fats that we turn into energy. About 1/3 of our energy needs should come from dietary triglycerols, like vegetable oils or butter or meat and things like that. Also, seeds that you use to eat, like beans or peas or peanuts, or pumpkin seeds. They contain a lot of fat. So usually, Seth just gave me some beans. These beans don't have a lot of fat in them, but the seeds that we eat, like our flax seeds and pumpkin seeds and things like that, they store energy for the new seedling before they germinate. And then once they actually become a plant, like behind me, you see like 160 plants. This is a marigold that's already flowered. I'm waiting for the sun to come out or the cold to break so I can plant some of these things out. They no longer rely on fat. They rely on photosynthesis, which is carbon dioxide, and then they store that as carbohydrates. But for us, 80% of our energy needs in our heart and our liver are met by the oxidation of fatty acids. So we already learned the structure of fatty acids, and we fully oxidize those carbon-carbon bonds, then we get energy out of that. Many hibernating animals, such as this grizzly bear here, rely almost exclusively on fat as their source of energy. So they spend a lot of time storing that fat, and then they can burn that fat slowly over the winter for energy. When you're running, if you're sprinting, first you need quick energy, so you turn to glycolysis in the citric acid cycle and break down glycogen because that gives you quick energy. But as we'll see, to break down fat, we have a lot of energy stored in our fat, but to break that down, it's a more complex process. So if you're running a marathon, then you turn to your fat reserves in order to get energy when your glycogen stores are depleted. Fats, in fact, provide very efficient fuel storage. There are advantages of fats over polysaccharides that we learned about before. Fatty acids carry more energy per carbon because they are more reduced. So that could be a multiple choice question. Fats are advantageous over polysaccharides because they carry more energy per carbon because they are more reduced. And they carry less water along because they are non-polar. So as we saw the very long demo before, they are non-polar, they are hydrophobic, and they are packed more tightly. Glucose and glycogen, which is a polysaccharide of glucose, are for short-term energy needs and quick delivery. But fats are for long-term, sometimes months, of energy storage, and they are good for slow delivery. Now we want to move on to our first objective, which is to learn how fats are transported and digested by the body. So when we need to use fat as a fuel source, we can get that fat in two ways, either just eating fat or by utilizing the fat that is stored in our fat cells. So the first thing we're going to talk about is how do we take the fat that we're eating and transport it throughout our body so that it can be turned into energy through the processes by which we catabolize fat. So let's imagine that you are right now eating, you're trying to be healthy, so you're eating some peanuts or some pistachios or something like that. And if we think about the structure of the fat that you're eating, we go all the way back to chapter 10, I think, and here is a triglycerol. So as you're eating those pistachios, you're eating a structure that looks sort of like this. Remember that we have a glycerol backbone. So that's a fat. It's part of our triglycerol. So our glycerol backbone. And it's attached to 3 fatty acids. So we have a fully saturated fatty acid chain. And here is another saturated fatty acid chain. And then here is a unsaturated fat. Alright, so glycerol backbone with fatty acids attached to it. They can be monosaturated, disaturated, or unsaturated, or fully saturated, unsaturated, or fully saturated fats. Okay, so we go along and we eat this fat, we ingest it, it goes through our stomach, it makes it into our intestines. So that's where we are now. We have these triglycerols, they made it into our intestines. And if you at all watch the little demonstration, we know that fats are not water soluble, so it's not easy for those water soluble globular proteins like lipases to break down that fat. So as your fat is sitting, these triglycerols are sitting in your small intestine. This is a triglycerol here, and then we have some just some fatty acids as well. You have bile salts that mix with those and emulsify those. So step 1 is the bile salts would mix with those and make sort of mixed micelles. So the bile salts are amphipathic, and it's like putting detergent and water and oil. It makes this heterogeneous, sort of homogeneous mixture of things where you have the fats mixed with the water. And in that way, when it's emulsified like that, then it can be acted on by intestinal lipases. So the bile salts mix with the digested triglycerols, forming these micelles. And then step 2 is the lipases can now act on them and break them down. And a lipase is just an enzyme that breaks down a lipid. That's what fats are, lipids. Okay, so once you have it broken down, it's going to be broken down into many different things. You'll have glycerol, you'll have the fatty acids, you might have one free fatty acid or a free glycerol, or maybe a glycerol backbone stuck to 2 fatty acid chains, any number of mixture of things. Well then, once we've broken those down a bit by the lipases, the free fatty acids can be taken up. So then the free fatty acids can be taken up by the intestinal mucosa. So then they're taken up by the lining of the intestine, and then they're actually repackaged. Now they're repackaged with cholesterol and other proteins. So they are free acids, but then they get taken up by the intestinal wall, and then they get repackaged with things like cholesterol, other transport proteins, so they can go through your bloodstream. So that would be steps 3 and 4. They get taken up, and then they get repackaged with other things like cholesterol, something called apolipoproteins, don't worry about the names of those too much, into something called plymomicrons. So I'll show you a picture on the next slide of a plymomicron. But just imagine that these triglycerols get repackaged with other stuff so that now that they can be taken and transported throughout the bloodstream. All right, so after they're situated into these chylomicrons, the chylomicrons can move throughout the bloodstream. So imagine now I have my fats that I ate, and now they're taken up, they're repackaged, they're broken down, and then they're built back up and repackaged in this thing called a chylomicron so they can go throughout the bloodstream. Once they go throughout the bloodstream, then they can be taken to where they are needed. So they can be taken to the muscles if you're working out, they can be taken to the heart, they can be taken to the brain or the liver, or wherever you need that fat to be broken down into energy. They are packaged in a way that they can actually go through the bloodstream and get to where they are needed. So once they are taken there throughout the bloodstream, then they can be converted back into fatty acids and glycerol. So now my fat is packaged in a chylomicron. It's taken to my muscles because I'm working out, I'm doing whatever I need to do. Then that triggers the release of a lipase, a particular type of lipase, which you don't need to know the name of. But that triggers the release of a lipase that can re-break down those fatty acids, or those triglycerols, into our fatty acids and into the glycerol backbone. And then we have ways where we can break down the free fatty acids and the glycerol into energy. All right, so just about four to six steps you have to be familiar with of how we get from the fact that we're ingesting fat and then turning those into a form that we can get energy out of those. So you eat them, they get broken down into free fatty acids and glycerol. By lipases, because you mix them with the detergent, our bile salts, and then they are repackaged with lipoproteins, other proteins, cholesterol, things like that, into this thing called chylomicrons. The chylomicrons can go through your bloodstream, and then when they reach where they need to be, like your muscle cells, then another lipase break those down again into free fatty acids and glycerol, and then those free fatty acids and glycerol can be turned into energy, and that will be the process that we'll find out in the next lecture. Let's talk a little bit more in detail about the particles or the proteins that actually help to transport the fat into the bloodstream or through the bloodstream. So remember that you ingest the fat, you eat it, there are bile salts that come in and break it down or put it into micelles, kind of repackage it so that it is more accessible for the lipases to actually break it down into free fatty acids and glycerol. But now once you have it in free fatty acids and glycerol, now they can be taken up by the lining of the intestines. Once they are there, they get repackaged into lipoproteins. So that's where we are on step maybe 3 and 4, where the fatty acids and glycerol get packaged with lipoproteins. So here, over here to the right, is an example of a lipoprotein. So a lipoprotein is composed of a lot of different things. It's just a pretty large particle. On the surface of it, we have phospholipids. So it has a phospholipid layer, just like a cell does, where we have our, phosphate group sticking out towards the water or the blood, and our lipid part that interacts with the inside. We will have cholesterols that might be interdispersed within the lipoprotein or on the surface of the lipoprotein. And our triglycerols that just got repackaged and stuffed into this lipoprotein would be in the middle. So those triglycerols can get transported through the bloodstream because they're packaged in something that has a layer of phospholipids that allows it to interact with the water in the cell. or in through the bloodstream. On the outside, we have these sort of carrier proteins, our lipoproteins. That means those are the ones on the outside that don't have lipids stuffed in them. So these are just going to be for cell recognition. They might bind to receptors. They are helping to carry this package of triglycerols through the bloodstream. So again, the lipoprotein is what encompasses this whole thing that has our triglycerols that we want to get through the bloodstream packaged inside. So our lipoproteins can vary in density with the amount of triglycerol relative to the amount of protein that we have. I mentioned before that we can have chylomicrons. So one of our lipoproteins that has a high fat density would be called a chylomicron. You've probably heard of these other things when you talk about good cholesterol and bad cholesterol. What does that all mean? It means that if you have a diet that's really, really high in fat, or you just ingested something that's really, really high in fat and you need to get that through the bloodstream, it might be packaged in a lipoprotein that's called a chylomicron. If you ate something that's a little less fatty, I guess, it would be packaged in a lipoprotein called a very low density lipoprotein. So very low density lipoprotein, VLDL. LDL is one you probably heard of. This is called the bad fats. So this one, when you have high cholesterol or a lot of the bad cholesterol, it means that you're probably eating a diet that's really high in fat and really high in cholesterol relative to the amount of protein that you're eating, because you have low, so I'll just put low density lipoproteins. And then if you're eating a diet that has a better ratio of cholesterol and fat to protein, then you would have high density lipoprotein. So the ratio of protein to fat is going to be higher. So this is usually what we think of when we think of good cholesterol or good levels. So all this means is that all of these are lipoproteins. They vary in density in the amount of triglycerols relative to the amount of protein. So lower density would be bad for you because it's higher in cholesterol and higher in fat. But high density would be better for you, I guess, because it's higher in protein and lower in fat and cholesterol. So we just learned what happens when you ingest fat and you need it to travel through your bloodstream to the cells that need them most so that it can be burned off as energy or metabolized. So what happens when you have energy needs and you want to use fat that is stored in your body? And so now we're going to talk about how we actually get the fat that's stored in our fat cells mobilized and carried throughout our bloodstream so that we can use those for energy. Alright, so first we're going to be utilizing the hormones epinephrine and glucagon. So if you're running a marathon or exercising or whatever, your body is going to first utilize the glucose that's in your bloodstream and the glycogen that's stored in your liver. Because that is really easy metabolizing for our short-term energy needs. But if you need more energy than you have available, your body is going to release either epinephrine or glucagon. So epinephrine gets released. We learned about the beta adrenergic receptors when you have the fight or flight hormone release. So you need energy now, something is happening where you need a lot of energy. This same epinephrine is going to activate lipases that are going to help us to break down fat. And also, when your glucose stores are low, you already used a lot of the energy you have stored as glycogen, your body is also going to release the hormone glucagon. Okay, so this is just going to be kind of a review from our signaling, our biosignaling. Here we have glucagon. So remember again, glucagon is released when your glucose stores are low, which you can use for energy, but now I need to use my stored fat as energy. So the glucagon binds to the surface of the cell on a receptor. It says glucose is low, so I need to burn fat now. It is a G protein coupled receptor, so we remember that it is attached intracellularly to a G protein. That G protein has a alpha subunit that dissociates, and it activates adenylyl cyclase. This is just a review from biosignaling. When you activate adenylyl cyclase, you convert ATP to our CAMP molecule. CAMP is a second messenger, remember? So CAMP is our second messenger that gets created. It allosterically modulates protein kinase A, so PKA. So what does PKA do? Well, it's a kinase, and kinases go around phosphorylating things, so it activates other proteins by phosphorylation. So now we just review, okay, glucagon says I need more energy, more than the glucose I have around. I activated adenylyl cyclase. I made some CAMP molecules. I activated PKA. So PKA, via our second messenger CAMP, is going to now activate two proteins, or two enzymes called lipase and paralipin by phosphorylation. So PKA phosphorylates paralipin and lipase, thus activating those two. Alright, so now we're at around step 3. If you can look at your figure, we activate hormone sensitive lipase, or HSL. So we call it here HSL. So that gets activated, and we can imagine if we activate a lipase and we want to break down fats, well, that lipase is going to break down the fat into a form that we can use either as free fatty acids or as glycerol, and glycerol can go into the glycolysis cycle. All right, but we'll come back actually to the hormone sensitive lipase, or the HSL. Let's talk about paralipin. You don't need to really know too many details about paralipin, but know that it is a family of proteins that sit on the surface of your fat cells. So our adipose tissue has all our fat stored in it, and those fat cells are surrounded by proteins that keep it from mobilizing prematurely. So you don't want the fat to start being broken down prematurely and going through the bloodstream before we need it. We want it to be stored. So the fat is surrounded by proteins called paralipin. So what happens when protein kinase A then activates or phosphorylates paralipin, then we have another protein that in turn it activates a lipase. You don't need to know the name of it, but I'll just say that it activates a lipase. Lipase gets activated. So we have an internal lipase that gets activated, and that's going to break down a triglycerol. So if you take off one fatty acid from a triglycerol, you get a diglycerol. And if you take off another fatty acid from a diglycerol, you have a monoglycerol. So paralipin gets activated. You activate another nameless lipase that breaks down the triglycerol into a diglycerol. The triglycerol turns into a diglycerol, makes a monoglycerol, and so then the monoglycerol can be broken down by our lipase that we just activated. So paralipin activates another lipase, an internal lipase. Triglycerol diglycerol, and then the monoglycerol can be broken down by our lipase that we activated by pKa. And so this lipase, I'll go to step 3, moves to the surface of the lipid droplet and cleaves the fatty acid and the glycerol, because that's as much as we want it broken down, or as much as the lipase can do. It can take that monoglycerol and convert it into a free fatty acid and a glycerol. So now we have the free fatty acids and the glycerol that are separated from each other, and they are taken up into the bloodstream and carried, because we can't just carry these greasy fatty things in our bloodstream, but they get carried up by another protein, serum albumin. So albumin is a protein, and it carries our free fatty acids. So it carries our free fatty acids wherever they need to go, to our skeletal muscles, to our heart muscles, wherever we need that fat to be broken down. It gets carried by serum albumin through our bloodstream, wherever it needs to go, and then it can be transported by a transporter into the cell, or fatty acid transporter. Alright, so very quickly, the steps are that we need we need energy, because we use up all our glucose. So glucagon binds the receptor. We then get CAMP. CAMP activates pKa. pKa phosphorylates and activates 2 different proteins, a lipase and a paralipin. Paralipin activates an internal lipase. The internal lipase breaks down the triglycerols into diglycerols. It breaks down the diglycerols into monoglycerols. And our lipase, activated by pKa, breaks that down into glycerol and free fatty acids. The free fatty acids can then go into the bloodstream. So they go into the bloodstream, but they have to be carried, because they're greasy, by another protein called serum albumin. The serum albumin can take it wherever they need to go, the muscle cells of the heart or whatever, and they can get transported into the cell by a fatty acid transporter. So we just saw how we break down our triglycerols into free fatty acids. So 3 free fatty acids. This is a triglycerol molecule here. Alright, so now we have 3 free fatty acids that can go through the bloodstream, carried by serum albumin, and then we have this glycerol. So let's talk about what happens to the glycerol first, because that's pretty simple. It's a 3-carbon sugar now, if you notice. Glycerol is a 3-carbon sugar. And first it's acted on by glycerol kinase. And what does the kinase do? It takes an ATP, our phosphate group off an ATP, and adds it on to our glycerol. So we have glycerol 3-phosphate. Then this is acted on by our dehydrogenase, which creates DHAP, dihydroxyacetone. So the glycerol gets phosphorylated, and then the glycerol 3-phosphate becomes dihydroxyacetone. Get out of my face. And we recognize dihydroxyacetone as DHAP, part of the glycolysis pathway. But of course, DHAP doesn't go through the glycolysis pathway. It gets converted by a triphosphate isomerase to our GAP molecule, which can then go through glycolysis. So in terms of what you should know about the fate of glycerol from our fatty acids, you should just know that it gets converted to DHAP, and you know if it gets separated to DHAP, it can go through glycolysis as GAP, which is a step you already know in glycolysis. Don't worry about this enzyme. or this enzyme here. Just that glycerol gets phosphorylated. Of course it's phosphorylated because it becomes DHAP, which you know the structure of, and DHAP gets converted to GAP, which goes through the glycolysis pathway. In terms of fatty acid transport, you should know that fats are degraded into fatty acids and glycerol in the cytoplasm of adipocytes. Fatty acids are transported to other tissues for fuel, but beta oxidation, that's the breakdown or the oxidation of the free fatty acids, occurs in the mitochondria. The small, if it's 12 carbons or less, fatty acids diffuse freely across the mitochondrial membrane. But what we're going to talk about next is how do larger fatty acids, which are most of the free fatty acids, we saw that they're 16, 18, or 20 carbons long, are transported through another process called the carnitine transporter. So before we can even utilize these free fatty acids for fuel in the beta oxidation of fatty acids, they have to get into the mitochondria. So we have to utilize a transporter to actually get them into the mitochondria, which is what we're going to talk about now. As we just saw, fats, or our triglycerols, are degraded into fatty acids and glycerol. And we know what happens to glycerol. It can go through the glycolysis pathway. But all of that happens in cytoplasm. But beta oxidation enzymes for fatty acids are found in the mitochondria. So we must shuttle the free fatty acids into the mitochondria. So as I just said, the small fatty acids that are 12 carbons or less can freely diffuse across into the mitochondria. But actually most of our fatty acids that we eat and that we store are more than 14 carbons. They're 18 or 20 carbons. And so they have to be transported via the carnitine transporter. This action takes place in 3 steps, which is called the carnitine shuttle. So carnitine is a molecule, and I'll show you that molecule on the upcoming slide. The first step is that we have to make an activated molecule. So this involves combining it with an ATP. So we have an ATP molecule. There are lots of ATP molecules around. Our fatty acid, so this here represents, with the R group, it represents our fatty acid that we just created. We broke it up into parts. We had to have a free fatty acid. And the carboxylate ion is adenylated by ATP. So we have this fatty acid gets adenylated by ATP. And so we have a free pyrophosphate now. This PPI is called pyrophosphate. And it's immediately hydrolyzed into 2 molecules of inorganic phosphate. And we've seen that those phosphates can be utilized in other places. And it's done with the help of an enzyme. It doesn't matter what the name of the enzyme is. It's a synthetase where we get a fatty acyl adenylate. Alright, so first we have to make a compound that's going to be able to be added to coenzyme A. So we have ATP in our fatty acid, and then we form a fatty acyl adenylate first. And then the next step, the thiol group of coenzyme A. So we've seen coenzyme A used a lot in other processes to get into the citric acid cycle. The thiol group of coenzyme A attacks the acyl adenylate that we just made, displacing the AMP. So now we've lost AMP and made a fatty acyl CoA. So we went from fatty acyl adenylate to a fatty acyl coenzyme A. So in the first step in the carnitine shuttle is to make an active compound that can be transported into the mitochondria, which is the fatty acyl coenzyme A. In steps 2 and 3 of the carnitine shuttle, we first swap out the coenzyme A for carnitine and then replace it with coenzyme A. So let's take a look at that very briefly. Just know the basic of the 3 steps of the carnitine shuttle. The first step we saw is to make the fatty acid into a fatty coenzyme A. Now we have this fatty acid, I'll circle it here, fatty acid coenzyme A on the cytosol, but we want it in the mitochondria. And so what we're going to do is swap out the coenzyme A for this carnitine molecule. It doesn't matter what the structure is. I know it kind of looks like an amino acid a little bit. So we swap it out, the coenzyme A, for a carnitine. And then that carnitine can pass through the intermembrane space and into the mitochondria. Once it gets into the mitochondria, then the carnitine is replaced with the coenzyme A. So all we've done is just a couple steps where we started out with coenzyme A attached to our fatty acid. We replaced with a carnitine so it can go through the transporter, the carnitine transporter. And then once we got over there, we have replaced and gotten back our fatty acid coenzyme A. We'll see in our next lecture what actually happens to that fatty acyl coenzyme A. But this is just an overview of what we're going to be looking into. We've gone through the trouble of breaking down our triglycerols. We got them through the bloodstream, and we now need to get it from the cytosol into the mitochondria so that we can then oxidize those fatty acids and get energy out of that. And that energy, most of that energy is going to be created in energy carriers through the citric acid cycle. So then what we'll see is 3 stages of fatty acid oxidation. The first stage is going to consist of oxidative conversion of 2 carbon units into acetyl-CoA and generation of NADH. So remember, we just made a fatty acyl coenzyme A. And if you remember, the acetyl-CoA goes into the citric acid cycle just as it did with pyruvate being converted into acetyl-CoA and then entering the citric acid cycle. So now we have 18, 16, 14, however many carbon fatty acid attached to a coenzyme A. We're going to take off 2 carbons at a time and create acetyl-CoAs, and acetyl-CoA can go into the citric acid cycle. And so that would be stage 2. And then, of course, stage 3 generates ATP from the NADH and the FADH2 that we created in the citric acid cycle. So the whole oxidation of the fatty acid is going to be creating acetyl-CoA molecules from however many carbons there are in the fatty acid chain. We'll pick up next time in the beta oxidation pathway, which is 4 steps: oxidation, hydration, and then another oxidation, and then acetyl transfer and release of acetyl-CoA so it can go into the citric acid cycle. So as we have our fatty acyl coenzyme A, it has to go through an oxidation, hydration, another oxidation, and release of acetyl-CoA so that we can go into the citric acid cycle and get energy. I hope you're doing well. I hope you're living well and seeking God in your isolation. Until next time.